Hello everyone, once again, I'm going to teach you about the mechanism of cell injury. There are six mechanism of cell injury. There are six ways by which cell enters into the cell injury or cell death. The number one is the ATP depletion. Number two is the damage to the mitochondrial membrane. Number three is the influx of calcium in intracellularly and loss of calcium homeostasis. And number four is the formation of reactive oxygen species. Fifth one is the defect to the cell membrane, and sixth one is the damage to the DNA. Now, this is a flow chart which shows the correlation between the basic mechanism of cell injury and the morphological features of the reversible cell injury and the irreversible cell injury. This is very good and very summarized flowchart which I have made it for you for your better understanding. And this shows this component this compartment shows the reversible cell injury, and this compartment shows the features of irreversible cell injury. What happened when there is ischemia? As you all know that oxidative phosphorylation occurs in the mitochondria, and you all know that oxygen is very essentially required for production of ATP, which occurs in mitochondria by oxidative phosphorylation. In case of ischemia, there is decrease in oxidative phosphorylation and there is decreased ATP formation. Because of decreased ATP formation, what will happen? There is decrease in the efficacy or function of the sodium potassium ATPase pump, and because of the decreased function or efficacy of this sodium potassium pump, there is influx of intracellular accumulation or influx of calcium in the intracellular compartment along with water and the sodium, and there is efflux of potassium out of the cell. What will happen? All these biochemical changes, what they will do, they will cause the cellular swelling. There is loss of microvilli, there is bleb formation, there is endoplasmic reticular swelling, and there is myelin figure formation. These are all these features which I have told you in morphology of reversible cell injury in cell injury portion. Why it happened, the cell injury, the cellular swelling, is because of the influx of calcium, water, and sodium in the cell. And there is also when there's oral cell swelling, obviously there is loss of microvilli, bleb formation, and there is overall cellular organelle swelling. Now, next thing we'll do, because of decreased ATP formation, there is increased anaerobic glycolysis. Because of increased anaerobic glycolysis, there is formation of lactic acid, or accumulation of lactic acid in the cell, and because of accumulation of lactic acid in the cell, there's decrease in the pH of the cell. And what we had learned in previous lecture of this cell injury, as you remember that there is clumping of nuclear chromatin. This is because of this decrease in pH, and because of decrease in anaerobic glycolysis, there is decrease in glycogen, and because of this decrease in pH, there is intracellular release and activation of lysosomal enzyme. Then cell will enters into the irreversible cell injury. Other effects because of decreased ATP production is detachment of ribosomes in on the endoplasmic reticulum. Because of detachment of the ribosome, there is decreased protein synthesis, and there is abnormal lipid deposition in the cell in the form of fatty change. Fatty chain is one of the morphological feature of reversibly injured cell, in which there is abnormal lipid deposition, basically triglyceride accumulation within the cell, and it look like clear spaces or signet ring appearance in the cell on HN hematoxylin and eosin staining. I told you that most important differentiating morphological feature in between the reversible cell injury and irreversible cell injury, the most important feature is the membrane injury. Membrane injury will differentiate. If the membrane is damaged, then cell will enters into the irreversible cell injury, and there is damage to the all cellular membranes of the cell itself and the organelles of the cell. And also there is leakage of intracellular enzyme. When there is damage to the membrane, also there is leakage of enzymes, intracellular enzymes to the extracellular compartment. What will happen because of membrane injury? There is intracellular release and activation of lysosomal enzyme. These changes because of membrane injury and also because of decreased pH in the cell. And because of these increased intracellular release and activation of lysosomal enzyme, there is a decreased basophilia in the cytoplasm, means there is more eosinophilic cytoplasm, more pinkish colorish cytoplasm, and there are nuclear changes like pycnosis, karyolysis, karyorexis, and there's protein digestion or protein denaturation in the irreversibly damaged cells. Let's discuss all these changes one by one. Now, number one is this is a flow chart which shows the combined combined pathway by which the ATP decrease in ATP, membrane damage, increase intracellular calcium, and reactive oxygen species cause the damage to the cell and the organelle membrane. Now let's discuss the first one, mechanism of cell injury, which is the ATP depletion. What will happen because of basically, as you all know, that the reduction in ATP level is a fundamental cause of necrotic cell death. The ATP depletion and decreased ATP synthesis are frequently associated with both hypoxic and chemical injury. The ATP is, as you all know, that the ATP is produced in two ways. The major pathway is oxidative phosphorylation, and the second one is the glycolytic pathway, which generate ATP even in the absence of oxygen using glucose derived either from body fluids or from the hydrolysis of the glucose. The major cause of ATP depletion are reduced supply of oxygen and nutrients, mitochondrial damage, and the action of some toxins. High energy phosphate in the form of ATP is required for virtually all synthetic and degradative processes within the cell. They include the membrane transport, protein synthesis, lipogenesis, and the deacylation and reacylation reaction for lipid turnover. What will happen because of ischemia? There is decrease in oxidative phosphorylation and decrease in ATP formation. Because of decreased ATP formation, there is failure of the sodium potassium pump, and there is influx of calcium, water, and sodium inside the cell, and there is efflux of potassium out of the cell. This results in the endoplasmic swelling, cellular swelling, loss of microvilli, and the bleph formation. As I have mentioned previously in flow chart, that there is increase in anaerobic glycolysis. Because of increase in anaerobic glycolysis, there is decreased glycogen, and there is decreased pH, which results in the clumping of nuclear chromatin because of lactic acid formation. The other defects are decrease in protein synthesis, and it ends in the lipid formation in the form of fatty change in the cell. Now, second one is the membrane damage, mitochondrial damage. The mitochondrial damage, as you all know, that the mitochondria are critical players in cell injury and cell death by all pathway. This should be expected because they supply life-sustaining energy by producing ATP. Mitochondria can be damaged by increased cytosolic calcium, reactive oxygen species, and oxygen deprivation. So they are sensitive to virtually all types of induce stimuli, including the hypoxia and the toxins. And what will happen because of increased cytosolic calcium, oxidative stress, and lipid peroxidation? There's decrease in ATP production, and because of decrease in ATP production, there's opening of this MPT pores, mitochondrial transitional pores, and there is a release of hydrogen ions and cytochrome C from the mitochondria into the cytosol. And these cytochrome C has a role in apoptosis, which we will discuss later on in my day 4 lecture, most probably. And also, along with the cytochrome C, there is a release of other pro-apoptotic proteins, which has also in function, which has also role in apoptosis. The third one is the influx of calcium. Third one mechanism is the influx of calcium. What will happen, as you all know, that the calcium ions are important mediators of cell injury. In keeping with this, depleting calcium protects cells from the injury induced by a variety of the harmful stimuli. Cytosolic free calcium is normally maintained at low concentration, means cytosolic calcium level is low as compared to the extracellular compartment. But when there is damage to the plasma membrane, the extracellular calcium, there is influx of extracellular calcium from extracellular compartment to the intracellular compartment. What it will do, this calcium which comes from the extracellular compartment to the inside the cell, it will activate different types of enzymes in the cells. There's also release of calcium from the damaged mitochondria, and there is also a release of calcium from endoplasmic reticulum from the damaged membrane of the endoplasmic reticulum. What all these intracytosolic calcium will do, they will activate variety of enzymes in the intracellular compartment. Like ATP will cause the hydrolysis of ATP and it will decrease the ATP. What will do the phospholipase? Phospholipase act on the phospholipids of the plasma membrane as well as the organelle membrane, and it will decrease the phospholipids, and what it will do, it will cause the membrane damage. What protease will do? It will cause the disruption of membrane and cytoskeletal protein. All these, they will cause the membrane damage. What endonuclease will do? If there's activation of endonuclease, as the name indicate, endonuclease, it will act on the nucleus. It will cause the nuclear chromatin damage. What will happen because of this all these repetition? What will do with the membrane damage? The membrane damage, the increased cytosolic calcium and the decreased ATP production. All these leads to the membrane damage. This shows the formation of free radicals by different ways, like UV light, ionizing radiation, smoking, inflammatory mediators, air pollutants. All these, what they will do, there's formation of free radicals. And what are free radicals? As you all know that the free radicals, they are reactive oxygen species and they are highly reactive. Ultimately, what they will do, they will cause the DNA damage in the variety of ways. That's it. These are references. From here I have made my lecture, and thank you very much once again. Hope you will like this video. Thank you.